In the second part of this lecture, we're going to talk about meiosis, the process that effectively enables sexual reproduction. So for the sexual reproduction to occur, one must have gametes. And each gamete must be a haploid cell, meaning that it contains only one set of chromosomes. Now, as we mentioned before, when gametes fuse, the zygote is formed. Zygote, unlike gamete, contains two sets of chromosomes. Each of the sets is inherited from the biological parent. So zygote is a diploid cell. So in order to produce haploid gametes, a diploid cell must reduce the number of chromosomes 2 times. In other words, in order to facilitate sexual reproduction, there should be a division that will reduce the chromosomal number set by half. So most animals, plants, and unicellular eukaryotes are diploid, meaning that the somatic cells in those organisms have 2 copies of each chromosome. Now these are called homologous chromosomes. In a pair, as you may remember, of homologous chromosomes, the chromosomes they match because these chromosomes contain the same genes in identical locations, genes that are called alleles. And as we mentioned before, one copy of each homologous chromosome is inherited from a biological parent, each biological parent. So here you can see that an egg or a sperm contain single set of chromosomes, and when they fuse, the resulting zygote contains 2 sets of chromosomes. And then adults are formed, the organisms are formed by cell division and mitosis, but then gametes are formed via meiosis, which occurs in testes in males and ovaries in females. So meiosis is a division of a nucleus in which haploid cells are formed from diploid cells. Now, in mitosis, you may remember that the number of chromosomes in parental and daughter nuclei are the same. It's the same ploidy level. So for instance, plants grow as diploid sporophytes by mitosis. And then they can also use mitosis to grow and produce eggs and sperm as gametophytes, although they are haploid. Now, meiosis is the process in which starting nucleus is always diploid, while the daughter nuclei are haploid. So basically, you replicate DNA once, one round of replication, and this round of replication is followed by 2 rounds of nuclear division. Major processes are designated as meiosis 1 and meiosis 2, and both of them consist of prophase, metaphase, anaphase, and telophase. Now here you can see that during interphase, the homologous chromosomes in the diploid cell are replicated, forming 2 chromosomes which consist of 2 sister chromatids. And then chromosomes are separated, and each of these homologous chromosomes is then further separated. The chromatids are separated, forming 4 cells, each with a single chromosome. So in the meiosis 1, just like in mitosis, the prophase 1 is preceded by an interphase. During interphase, in first gap stage, cell accumulates resources for DNA replication. In S phase, each chromosome is replicated, just like in mitosis. And now it consists of 2 identical sister chromatids. Now, chromatids are held together at the centromere by cohesin protein. So far, it's very similar to mitosis. During early prophase, these homologous chromosomes are attached at the nuclear envelope. And when nuclear envelope breaks, the homologous chromosomes form a tetrad, a pair, using so-called proteins of synaptonemal complex shown here in this white fibers. So when two homologous chromosomes tightly pair, it's called synapsis. During synapsis, the corresponding alleles of the same genes are aligned with each other. Now, synaptonemal complex, which is a bunch of proteins basically, supports the recombination, the exchange of the segments of these chromosomes between homologous non-sister chromatids. It's called crossover. The crossover can be observed as chiasmata, which we're going to talk about. Now, this is true for 22 pairs of homologous chromosomes that are not sex chromosomes. X and Y, two sex chromosomes, aren't completely homologous, but they share some homology. So in prophase 1, X and Y are paired via so-called partial synaptonemal complex between the regions that are actually homologous to each other. So now let's take a look at the very interesting phenomenon called crossover. So during crossover, special recombination nodules, protein complexes, are formed along the synaptonemal complex. They mark the points of chiasmata to mediate the crossover between homologous non-sister chromatids. So essentially what happens during crossover is the exchange of the equivalent DNA parts that contain the same alleles, between the chromosomes that are derived from an egg or from a sperm. During crossover, the double-stranded DNA in each chromatid is cut. The ends of the cut are modified, and non-sister chromatids connect. Now, synaptonemal complex breaks then, and chromosomes condense, being attached at the centromere and at chiasmata. Each chromosome has at least one chiasmata, but often there are more than that. And when you have this arrangement, this two homologous chromosomes, each consisting of four sister chromatids lying along each other, call it the tetrad. So once the crossover, the recombination is completed, each of this each of these homologous chromosomes now have slightly altered genetic sequence. So in other words, each homologous chromosome now contains the combination of the genes from the mother and the father, the combination that did not exist before the crossover. So crossover is essentially the first source of genetic variation that meiosis produces. It can occur virtually anywhere along the synapsed chromosomes, and you can imagine how many different variations that can produce, resulting in recombinant chromatids with varying maternal and parental gene combinations. Now, in prometaphase 1, the spindle fiber microtubules attached to the kinetochore proteins at the centromeres. So microtubules grow from so-called microtubule organizing centers, or MTOC. In animal cells, its role is played by centrosomes. So from each pole, microtubules that extend from each pole attach to one of the fused homologous chromosomes, not chromatids, the entire chromosomes. So each chromosome of the pair attaches to a microtubule from the opposite pole of the cell. It's similar to the attachment of sister chromatids in meiosis, but in this case, it's homologous chromosomes. So at the end of prometaphase 1, each tetrad is attached to microtubules that extend from both poles. One homologous chromosome in a pair is attached to microtubules from one pole, and the non-homologous chromosome from that pair is attached to the microtubules extending from another pole. Now, these homologous chromosomes still held together at chiasmata and at the centromere. During metaphase, the homologous chromosomes randomly align at the metaphase plate. Now, this is the second source of genetic variation during meiosis. It's called independent assortment. So each chromosome, whether it is maternal or paternal, can potentially face either pole of the cell independently of others. So the arrangement of each pair does not depend on the arrangement of other pairs. So this random alignment of tetrads results in a great variety of combinations of chromosomes and gametes. So two sources of genetic variation in a cell are essentially crossing over that occurs during prophase 1 and independent assortment that occurs in metaphase 1. So 23 human chromosomes, that's a bit of a typo. It allows for 2 in the power of, I think, 23. So more than 8 million possible combinations of chromosomes and gametes. You can imagine how diverse the population of gametes can be. So both crossover and independent assortment result in a phenomenal genetic diversity, ensuring the unique composition of the genes in each gamete. So this image shows you the separation of independently arranged homologous chromosomes, and you can see that even having two homologous pairs can serve as an example of the genetic variability that the cells can possess. Now, anaphase is pretty straightforward. During anaphase, the microtubules pull the homologous chromosomes apart towards the opposite poles, but the chromatids stay connected at the centromeres. Chiasmata, the points of crossover, break when chromosomes separate, obviously. In telophase, chromosomes arrive at opposite cell poles, decondense, and nuclear envelope forms around the set of chromatids. In animals and most fungi, cytokinesis occurs through the formation of cleavage furrow. The actin ring, the ring formed of protein actin, contracts and divides the cytoplasm. In plants, the cell plate is formed. Cell plate is formed from Golgi vesicles at the metaphase plate. So Golgi vesicles, they crowd around the metaphase plate, forming the cell wall, which will then separate daughter cells. So at the end of the meiosis 1, you have two haploid cells. Each cell contains one set of chromosomes. If it would be humans, 23. Each cell has a homologous chromosome from each pair, from each of 23 pairs. And each chromosome still has 2 sister chromatids. Sister chromatids will be separated in the next division, meiosis 2, which will result in 4 distinct haploid cells. Between meiosis 1 and meiosis 2, there is often interkinesis, a phase without DNA replication. It prepares cells from meiosis 1 to enter the meiosis 2 at the same time. Meiosis 2 effectively separates sister chromatids in each haploid cells produced during meiosis 1, resulting in 4 genetically unique gametes. It very much resembles mitosis, but the number of chromosomal set, the number of chromosomes is just a half of the diploid set that is separated during mitosis. So in prophase 2, chromosomes condense and nuclear envelopes break down. Microtubules are extended from microtubule organizing centers, which are moving to the opposite poles of the cells, and the new spindle apparatus is formed. In prometaphase 2, the spindle apparatus is fully assembled, and each sister chromatid is attached to microtubules from opposite poles through the kinetochores. So in metaphase 2, you can see that sister chromatids align along the equatorial plate, fully condensed. Each sister chromatid is attached to kinetochore tubules at the centromere, the kinetochore. And just like in mitosis, during anaphase 2, kinetochore microtubules, they pull chromatids to the opposite poles of the cell, while non-kinetochore microtubules extend and stretch the cell, preparing it for cytokinesis. In telophase 2, chromosomes that are at this point, the poles of the cell start to decondense. The nuclear envelopes are formed around each set of chromosomes, and then in cytokinesis, cytoplasm in each case divides, which results in 4 unique haploid cells. Now, if you look at the composition, genetic composition of the cell, each gamete, each cell, each haploid cell is genetically distinct because of the independent assortment and the crossing over. So this table provides you with a summary of meiosis 1, and meiosis 2. I have no intention of going over, but it's a really good summary with illustrations and what happens at each stage. Now, if we try to compare mitosis versus meiosis, there are some similarities, and there are also some differences. So both mitosis and meiosis involve nuclear division in eukaryotes. Both have prophase, metaphase, anaphase, telophase. Both cases the spindle is formed, chromosomes are aligned at the equator. But the differences is that mitosis is a single division, which produces 2 genetically identical nuclei, and usually 2 cells. Meiosis, 2 divisions, producing 4 nuclei that are genetically unique and are inside usually 4 cells. During mitosis, the original chromosomal set is retained. If haploid cell undergoes mitosis, then 1 set. If diploid cell undergoes mitosis, then 2 sets. Meiosis, on the other hand, halves the chromosomal set, which results in haploid cells, and each of them has only one set of chromosomes. It's called reductional division. There are some features of meiosis 1 that are not present in mitosis. For instance, formation of synaptonemal complex, chiasmata, and crossover, which essentially facilitates the genetic exchange between homologous non-sister chromatids. Now, during meiosis 1, homologous chromosomes, not chromatids, are pulled to opposite poles, which reduces the ploidy of a cell from 2 sets to 1. Meiosis 2 is much more similar to mitosis, because both processes involve separation of sister chromatids. When chromosomes align at the metaphase plate and kinetochores pulling sister chromatids to opposite poles of the cell. So meiosis 2 essentially is not a reduction division, because it keeps that one set of chromosomes that it started with. Now, there are several reproductive strategies that living organisms use. One of them is asexual reproduction, such as one seen in bacterial cells or prokaryotic cells. What is the advantage? First of all, it's efficient. Offspring or genetic clones of the parent, and unstable environments where parents are evolutionarily successful, that genetic identity can be really advantageous. Another advantage is that asexual reproduction does not require a partner, whether it is budding, fragmentation, spore, or asexual egg production. It is useful for organisms that are solitary or exist in favorable conditions. The populations that use asexual reproduction grow faster, since all individuals in the population can reproduce, not just females. Disadvantages. First and foremost, limited genetic variation, which can arise through mutations, and in the case of bacteria, horizontal gene transfer. Mutations occur randomly, and basically one of them, it's kind of a major source of genetic diversity in asexual populations. Now, that limit in variation may reduce the adaptability to changing environments and may reduce the long-term survival for that population. Contrary to this, during sexual reproduction, genetic diversity is incredible. So genetic variation is achieved through several mechanisms. First of all, is crossing over, which results in the new gene combinations, and independent assortment, which results in the unique set of chromosomes and gametes. The genetic diversity of the offspring significantly increases the presence or the emergence of genes that are advantageous for survival, giving those populations greater reproductive success and increased population growth. Now, mutations can and will be mixed and reshuffled in each generation because of the crossover and independent assortment, and it allows for adaptation while the beneficial gene combinations are maintained. So, as we mentioned already, this genetic diversity increases the resilience to environmental challenges, and in the long term, sexually reproducing populations have a survival advantage over asexual population. For some reason, there's a double here. So now we're going to talk about different life cycles that are characteristic for different groups of organisms. So first is diploid dominant life cycle. In this case, meiosis and fertilization are two alternating processes. The events between fertilization and meiosis may be different based on the reproductive strategy of the organism. Now, just a reminder that meiosis reduces the number of chromosomes by half, and results in haploid cells production, while fertilization includes the merge of two haploid gametes and the restoration of the diploid state. Animals use diploid dominant life cycle. In this case, they have multicellular diploid stage that is visible and dominant. The only haploid cells that are produced are gametes. So gametes are produced from germ cells, which are generated within the gonads. Testes in males and ovaries in females. Germ cells divide mitotically to maintain the cell line and use meiosis to produce haploid gametes. Gametes fuse during fertilization, forming a zygote. So there is no multicellular haploid stage. Fertilization is required to restore the diploid state when gametes fuse. Now, in haploid dominant life cycle, which is characteristic for fungi and some algae, the dominant multicellular stage is haploid. So haploid cells here, they form the main body of the organism. For instance, mycelium of fungi. Now, in order to form these multicellular tissues, the process of mitosis is utilized. Mitosis can be used for both haploid and diploid cells. Now, during sexual reproduction, haploid cells from the mating types, plus and minus, because they're really like not female and male, but two different mating types, they combine, forming a zygote. Diploid zygote undergoes meiosis, which generates 4 haploid spores, in which the genetic material is combined now. Spores thrive in favorable condition, forming haploid structures, which will produce a zygote during the nuclear fusion, and the cycle starts over. So you can see that most of the events in haploid dominant life cycle happens during the haploid phase. A lot of plants and some algae use alternation of generations type of sexual reproduction. In this case, the life cycle includes both haploid and diploid stages. So a gametophyte, which is haploid, produces gametes without meiosis, because it doesn't require meiosis, it's already haploid. So there's an egg, there's a sperm. An egg and the sperm fuse during fertilization, combining and forming diploid zygote. The zygote undergoes mitosis, developing a multicellular sporophyte. Special cells in sporophyte undergo meiosis, cells in sporangia, producing spores, which are haploid. Spores develop in gametophytes through the mitosis, and the life cycle is now complete. So this page simply summarizes 3 different strategies of sexual reproduction that you must know. Next time, we're going to talk about genetics.